I mentioned in the last video that something really interesting happens during prophase 1 of meiosis 1. And that is called synapsis and crossing over. So one of the key events that happens during prophase 1, now remember this is after DNA replication. This is after the chromosomes have condensed and become visible under a microscope. And the first thing that happens is called synapsis, and that's when homologous chromosomes pair up. So what do I mean by homologous chromosomes? Well, remember, in organisms that reproduce sexually, they get half of their chromosomes from dad and half of their chromosomes from mom. So if we're just talking about chromosome number 1 in humans, for example, after DNA replication, chromosome number 1 from dad is going to look like that, and chromosome number 1 from mom is going to look like that. Well, why is it important that homologous pairs of chromosomes line up? Because remember, each chromosome number 1 has the same collections of genes on it. Okay, they may be different alleles, they may be different versions of those genes, but they've got the same collection of genes. So if there's a gene that determines skin color on chromosome number 1 from dad, then there's going to be a gene that determines skin color on chromosome number 1 from mom. Okay, in the same location. I think this is another vocabulary word that maybe we didn't do in the first video, but a locus on a chromosome is a gene location. Okay, so if the locus for a melanin enzyme or a gene that controls melanin production is on chromosome number 1, it's going to be on chromosome number 1 in the same location from mom and from dad. Okay? All right, so during synapsis, homologous pairs of chromosomes pair up, and they don't line up side by side. Okay? It's easier to draw it that way, and so sometimes you're going to see homologous pairs of chromosomes drawn side by side, but in fact, they are lining up all along their length. You can think of it more like the two pieces of bread in a sandwich. Okay, they are laying on top of each other. They are sandwiched together, and so each chromatid is aligned with the chromatid from the other homologous chromosome. So if you look over here, in the figures from the book, you'll see that the blue chromosome is a pair of sister chromatids and the red chromosome is a pair of sister chromatids, and those two chromosomes are aligning all along the length of both chromosomes. That is synapsis. Now, that may seem kind of mundane, but I want you to think about the kind of feat that is. That in a teeny tiny cell, in a teeny tiny nucleus, in humans, the 44 autosomes are going to find their appropriate partners and form 22 homologous pairs. Now, how they do that is still somewhat under investigation. There are some things that are known about how the homologous chromosomes find each other, but not completely. But we know that it happens, and we know that it happens faithfully, that the pairs are faithful to each other. You're not going to have a chromosome 1 pairing up with a chromosome 14. Okay, so those homologous pairs find each other. Well, why do they have to do that? They do that in order that crossing over can take place. All right, so crossing over is when non-sister chromatids swap DNA. They swap genetic information. And what I mean by that is that the chromosomes, the non-sister chromosomes, are physically broken, and the non-sister ends are rejoined together. And so the result is that you're going to have chromosomes now that have different combinations or potentially different combinations of alleles. Okay? So, for example, if on these chromosomes, let's just say, what's a human trait that we know has a simple dominant recessive relationship? Let's just say that we're talking about the ability to roll your tongue, okay? Tongue rolling is dominant, so big R would be someone who could roll their tongue. Little r would be a non-roller, okay? Well, let's just say that the chromosome, the original chromosome that this individual got from dad, had two big R's, and the chromosome that originally came from mom had two little r's. So mom was a non-roller and dad was a roller. And let's just play pretend that on this same chromosome, it's not actually, but we're just pretending here, that on this same chromosome, the gene that controlled whether or not someone had a widow's peak, which is when your hair grows in that V on your forehead, on the top of your hairline, was on the same chromosome. And so dominant is the presence of the widow's peak and recessive is no peak. So let's just play pretend that dad also is dominant for having a widow's peak and mom does not. Okay? W's are kind of hard to tell, capitals and lowercase. All right, well, before crossing over, both dominant alleles and both recessive alleles were on the same chromosome. In our made-up example, after crossing over, we're going to have chromosomes now that have the dominant tongue rolling allele on the same chromosome as the recessive widow's peak allele, and the recessive tongue rolling allele on the same chromosome as the dominant widow's peak allele. So the result of crossing over in our made-up example is that we have shuffled the alleles for tongue rolling and widow's peaks, and we have different allele combinations where that crossing over took place. So the result of crossing over is increasing genetic variability because by the time we get to the end of meiosis and we have those 4 haploid cells, the chromosomes in these haploid cells are going to look different than the chromosomes did in the parent cell. And remember, these daughter cells, at least in animals and most plants, these daughter cells are gametes. They are sex cells. And so if one of these gametes happens to be involved in fertilization, there's going to be an organism that develops as a result of that fertilization, and that organism is going to be genetically different than its parent. Okay, so remember, in mitosis, the outcome of mitosis is genetically identical daughter cells. In meiosis, the daughter cells are genetically different from the parent. Crossing over is one of those reasons that the daughter cells are genetically different than the parent. There are additional reasons that we're going to talk about in the next lecture. Okay, now another thing that I want to point out is that each pair of homologous chromosomes undergoes crossing over. So in this figure, we're only showing, for simplicity, one homologous pair of chromosomes. But remember, in humans, there are 44 chromosomes, so 22 homologous pairs. That's a lot of crossing over. That's a lot of allele combination shuffling, okay? And there can be more than one crossing over event per homologous pair per round of cell division. So it doesn't just have to look like the examples that I've drawn and the examples in the book. There can be multiple crossing over events that take place. And we only showed the crossing over taking place on one set of non-sister chromatids. Make that a little bit bigger. On one set of non-sister chromatids, but because, remember, those homologous pairs of chromosomes, they're sandwiched with each other. Both sets of non-sister chromatids can have crossing over going on. So that's a lot of genetic variability that can be increased as a result of crossing over. And crossing over only occurs during prophase 1 of meiosis 1.